not showing in this, no? Yeah. The display is not coming. Yeah, it's visible. Okay, good afternoon all. Hope I'm audible to you. I'm Lakshmi Kantan. I'm working as a senior marketing manager. I'm from Everlife CPC Diagnostics. This Everlife CPC Diagnostic have been in this industry for almost 4 decades, and as well they are the market leader in autoimmune segment, which you know very well. And other than that, we have other diagnoses like allergy and other things also. So when it comes to this autoimmune diagnosis, this indirect immunofluorescence, it plays a major role. In today's session, we will be discussing about the procedure. We are having the hands-on also, and followed by we'll be having the blood system, which is how it is working. We'll show that on also, and other than that, we'll be seeing some of the patterns also. So these are all the major things which we are going to cover in next 2.5 hours. Okay, we'll keep you engaged by showing the patterns and as well as the procedures also. Yeah. So this is the prevalence of ANA all over the India, world. So it is increasing day by day in all the places. Since these are all the basic, I'm not touching so much in this. I'm going to the procedure in next 2-3 minutes. So majorly, if you are talking about the autoimmunity, the immune response, uh, you know, to the self tissues. The major reason behind it, there are multiple reasons behind it. We can say genetical reasons, and other than that, we have infection, toxic chemicals. There is an imbalance in the gut microbiome. So put together all the factor, it contributes to autoimmunity. It can affect the single organ or it can affect the entire system also, which we know very well. Systemic autoimmune rheumatic diseases. When it comes to systemic autoimmune rheumatic diseases, it may cause organ failure, it may cause disability, or it may cause premature mortality also. Loss of the function of the particular organ also it may happen. But when it comes to the prevalence, it is more in female gender and okay. So it is because of the XX chromosome which we are talking. Because of this, autoimmunity is chances are more in female gender. I'm going to the autoimmunity and as well as the prevalence of the diseases when it comes to anti-nuclear antibody. If you are talking about anti-nuclear antibody, the prevalence, it is more with the systemic lupus erythematosus, which is 95 to 100%. If we are talking about drug-induced, it is 95 to the least chances if we are talking ulcerative colitis, which is 26%, and other than that, if you are talking about other rheumatic diseases and rheumatoid arthritis, it is 20 to 40% only. But rest of the diseases, if we are talking, which is including Sjogren syndrome, which is 70 to 80% of the ANA prevalence, which is holding, and other than that, mixed connective tissue disorder and drug-induced lupus, it is having 100% prevalence over ANA, anti-nuclear antibodies. So these anti-nuclear antibodies are nothing but the antibodies are produced against the nucleus of the cells. Other than that, in recent days, we have the antibodies against the cytoplasm also that we will discussing in the upcoming slide. The major parts of the antigen which is acting again, producing antibody are like DNA, histone, we have ribosomal B protein, and other than that, we have SSA, Sjogren syndrome A and as well as Sjogren syndrome B. Various antigens are available that we will discuss in the upcoming slide. But anyway, the diagnosis, it plays a major role in this, and it holds maximum, diagnose maximum number of autoimmune diseases in this ICAP. It's nothing but international consensus on ANA pattern. They have given the terminology which is known as anti-nuclear antibody. But are we detecting the antibody with the nucleus alone? No. We are detecting the antibody against the cell also. So recently we have changed this into an anticellular antibody. Finally, the final proposed term is HEP2 cells, human epithelial cells. So when compared to normal cells and as well as human epithelial cell, the more number of mitotic cells are present in this. So that is the reason we are pronouncing this. Let me come to the next one. The each and every antigen and as well as the prevalence are mentioned here. So earlier we discussed about ANA, and other than that now we are coming to the antigen level. If you are talking about the antigen level, the dsDNA systemic lupus erythematosus, it is 60 to 90%. If you are talking about ssDNA, multiple autoimmune disorders are included in this. And other than that, if you are talking about UNRNP, again multiple connective tissue disorders are available. So these are all the individual antigen which is for autoimmune disorders, which we will discuss it by using the patterns in the upcoming slides. I'm not touching the slides too much, but have only one thing in your mind, that the prevalence is more with these antigens. And other than that, if you're talking about drug-induced SLE, systemic lupus erythematosus, majorly procainamide, hydrolysine, isoniazid, minocycline, and as well as infliximab, which induces systemic lupus erythematosus, which comes under drug-induced SLE. Yeah. So these are all the parts of the cells which we know very well. So each and every cell, it holds a different functions, and where we are targeting right now, it is nucleus. And other than that, the parts of the cytoplasm also, if the antibody is targeting the antigens against the part, then we can have the particular syndrome over it, that we'll discuss in the upcoming slides. I'm not touching, these are all the very basic things. Okay. Coming back to the ANA screening, if you are discussing about this ANA screening, as per this American College of Rheumatology, they have recommended that IF indirect immunofluorescence is the gold standard for the screening of autoimmune diseases. Like, so what is the next one in case if we are detecting through IF? First, screen any kind of autoimmune disorder through indirect immunofluorescence technique, and other than that, go for the blot in the next step. In case if you are going for the blot or ELISA in the first step, I can say many of the healthy people will be having antibodies against the normal antigen ANA also. So that is the reason, always go for indirect immunofluorescence technique first, then followed by either immunoblot or we can go for ELISA also. Okay, what's the difference between direct immunofluorescence and as well as indirect immunofluorescence, which we biochip technology and as well as tighter plane technology. So in this biochip technology, you can see the tissue is separated, and as well as this tissue are cut in a different form. It pasted in a biochips. Okay. So what are all the things can be pasted in this? Majorly the tissue section, purified antigen, cultured cell line will be pasted in a single biochip itself. Okay. What is the advantage of this one? So when I'm having this cell line in a single biochip, what I will do is I can paste the antigen, either the organ tissue I can use, or I can use the different tissue form, and other than that any other antigen also I can code. For example, if you are talking about the ANA basic one, it is 2 biochip technology. So in this 2 biochip technology, it is having the tissue and followed by the primate liver only. Okay, when I want to code the organ, I can go for 3 biochip, 4 biochip, 6 biochip, and different antigens can be pasted in that. For example, so in a single biochip, I can paste in the 6 biochip, I can paste organ tissue here, recombinant antigen here, cultured cells here, bacterial or unicellular organism or other fixatives can come in a single biochip itself. Yes, madam. No, madam. In this one, for example, we are having 10 different one sample can be tested with different antigens. Yes, madam. Yes, madam. So this biochip is also available. So this is the way it is coming. So we are coating 6 different antigen in a single biochip itself, but when it comes to ANA, it is only 2 biochips. One is hepto cells and another one is primate liver. With the hepto cell itself, we can find out multiple number of antibodies that we will discuss in the upcoming slides. Am I little fast? Okay. Okay. So why we are using this Hep210 cells here? So if we are talking about this Hep210 cells, this Hep210 cell, multiple mitotic cells in this. So when it comes to normal Hep2 cell, it is having multiple dividing cell in it, so that the interpretation will be better in it. So that is the reason we are having this patented technology of Hep210 in biochip. Okay. When we are talking about the advantages of the biochip, which I have discussed with you, when I want to discuss, the multiple antigen for a single patient, it is possible. This is the first thing. The second thing, if you want to test multi-patient, that is 10 patients in a single time, I can test in a single slide itself 10 patients. So those kind of advantages are available in this. So that is known as multiplex technique. So we have different pack sizes for biochip. For example, 5 patient slide, and we have 10 sample slide also. So we can process in this way. I repeat once again, in a simple thing, I can have multiple antigen for a single patient, and I can have 10 patient, I can run multiple patient in a single slide also. So this is the biochip technology. I'm going to the tighter plane technology, next one. Okay, so let me explain you about this tighter plane technology. For example, in the normal procedure, what we will do in the indirect immunofluorescence, we will add the substrate over the microscope slide, and for incubation we'll use the humidity chamber. So because of that, what are all the disadvantages we may have? The different start times for the different samples. For example, one sample I'll add at one time, and for the next sample I may take time. So uniform incubation will not be available for this basic technique. And other than that, the humidity chamber and risk of cross contamination, all those things are there with this technique. So that is the reason we are going for tighter plane technology. So when it comes to this tighter plane technology, the reagent tray, we will apply the sera, and other than that, rather than adding the sample directly to the slide, we will add to this biochip directly. Then we will place the slide over it. So when we are placing the slide over it, the incubation will be in the uniform time, And other than that, if you are talking about the slides also, the evaporation chances are very, very less. So we need not to go for the humidity chamber basically. So this is the tighter plane. So during the hands-on, you will see this tighter plane. It will be having hydrophilic layer and as well as hydrophobic layer. Madam, you can see this without touching this. Yeah. Okay, so this hydrophilic layer, where you will add the sample, and hydrophobic layer, it will not go away from that. So that when I apply that slide above that one, the sample will go directly to the slide and the reaction will start at the uniform time. So when I want to add multiple samples, for example, when I want to add 3 samples, that reaction will happen at the same time. The incubation time will be same. So that one, during the hands-on, we will discuss. And other than that, cross-contamination will not happen. One sample to another sample spillover will not happen because of this hydrophilic nature. Okay. So this is the, so this is the starting dilution for ANA. When it comes to ANA, and if you are talking about ANA, the starting dilution is 1:100. So always we recommend 2 dilution. Go for the 1:100. The weakest one, go for the maximum positive one. So these 2 dilution, when you go for it, we can come for the titer understand the titer in a better way. So that is the first thing. So what is the dilution to be done for indirect immunofluorescence technique for ANA and as well as ANCA and other profiles? So let's see one by one. If you are talking about this one, when it comes to the group 1, you can see the group 1, where is the positivity it starts? It starts from 1:10. And the strong positivity, it is available in 1:100. So what is the group 1? We can say this group 1, which is including ANCA antibodies, dsDNA. So for this ANCA antibody and dsDNA, the starting dilution should be 1:10. Okay, when we want to go for ANA, ASMA and AMA, the starting dilution should be 1:100. So please remember this, ANCA and dsDNA, the starting dilution is 1:10. And if you are talking about ANA, ASMA and AMA, the starting dilution is 1:100. Okay, what is the antibody titer? The weaker one is the antibody titer. So what is to be considered as antibody titer is weakest one. For example, let me take this one. The fluorescence at 1:100 and 1:1000, the antibody titer is the weakest one, which is 1:100. Okay, if you are talking about the next antibody titer, 1:100 is strong, 1:1000 is negative. Then what is the weaker one? What is the antibody titer? Okay, I'll explain you again. The dilution, it starts from 1:10, 1:32, 1:100, 1:320, followed by 1:1000. So the first titer, 1:100 was weak, 1:1000 was negative. So I consider weak as an antibody titer. So which one is antibody titer here? 1:100. Okay, when it comes to the next one, the stronger one is 1:100, the negative one is 1:1000. What is in between 1:100 and 1:1000? Yes, madam. Okay, I'll explain you in this way, madam. So when you are starting the fluorescence, when you are starting the initial fluorescence, so that is the weakest one. Okay, when you are going for the next dilution, after dilution, you will be having the strong fluorescence. Okay, so the titer for this treatment, other things to be considered as weaker one only. Okay, let me explain the second one. So 1:100 is strong, 1:1000 is negative. So that means it is in between. So we cross the weaker one also. The crosser would be, that is antibody titer is 1:320. I'm coming to the next one. 1:100 is strong and 1:1000 is weak. Which one is antibody titer? 1:1000 only. Because always consider the weaker one as the antibody titer. Sorry, sir. No, sir. Based on this dilution, actually it has to go in this way: 1:10, 1:32, or after that 1:100, 1:320, and 1:1000. So this is the way the dilution has to happen. One by one, sir, please. Yeah, we have to go for the weaker one. We will have to select the weaker one as a titer. Yes. Yes. So where the immunofluorescence is weaker, that one to be considered as the titer. Weaker one is to be considered as a titer. Okay. I'm showing the procedure right now. Please pay attention. After this, we'll go for the hands-on. During the pattern session, we'll go little slow. Don't worry about it. Right now, I'm touching the basic. That is the reason we are running fast. See this? So in this procedure, we are diluting the sample. What is the dilution we mentioned earlier for ANA? 1:100. So when it comes to ANCA, it is 1:10. So 1:100, I'm taking 1 microliter of the serum, and other than that, I'm adding 100 microliter of the PBS solution. So we will tell you what is PBS solution and all those things during the procedure. So we have prepared the dilution right now. Going to the next step. So as I told you, the titer plane also, which I have shown you, we are adding we can add the positive control here and negative control here, and positive control here and negative control here, followed by the sample of the patient sample we will add. So this is only titer plane. We have not kept the slides over it. So what is the next step? We are placing the steps over it. So the reagent of the sample, it will be absorbed by the slides. Then the antigen and antibody reaction will happen. It will be for 30 minutes. The incubation 30 minutes to be completed. After that, what I need to do, I need to flush once. I should not repeatedly wash it. I need to flush once. Once I flush the solution, I will soak it in the PBS solution for 5 minutes. So what is the next step? To add the secondary antibody. As I told you, primary antibody is our serum, secondary antibody is conjugate. We need to wipe the slides over the edges only. Do not touch the biochip. Do not touch the biochip, and we are placing over it for 30 minutes incubation. Again, remove that slide, flush with the PBS solution, and soak it over the Copeland jar. Then wipe it again. Keep the glycerol, and you can see under the microscope. So this is the way the procedure is happening. Any doubt in this procedure? During the discussion, we can discuss. 2 times 30 minutes incubation, madam. And for soaking, we need have 5 minutes. Okay. So overall, as of now, we discuss about the prevalence of ANA all over the world, and we discuss about the guidelines of ANA. When we are discussing about the ANA diagnosis, there are multiple autoantibodies present in it. Among that, you can say the nucleus antibodies, it plays a major role in autoimmune diagnosis. Okay, I'm coming to the next one. When it comes to the diagnostic guideline, which is given by American College of Rheumatology, go for indirect immunofluorescence technique, which is a gold standard for primary diagnosis, which will be having 100+ antibodies over it. So the next thing, once you complete that, for confirmation, go for blot or ELISA. So this is the way we can go for it. And as well as right now, we discuss about the, how the tests are being done. Okay, there are multiple stages of cells. Please don't, which we know, cell cycle. I'm not going to focus on that. Let's see the fluorescence alone. See the fluorescence. This is the, I'm highlighting this picture. Please see this. The first one, the fluorescence of the chromosome during the mitosis phase. And the next one is fluorescence of the cytoplasm during the mitosis phase. Okay, here we can see maximum number of nuclei are positive. The fluorescence are seen on the nuclei, but in the other case, the nucleoplasm will be positive. Okay, so the nuclei will be negative. So that we will see in the upcoming slides one by one. And other than that, for this indirect immunofluorescence, we are having this microscope. This microscope, let me make it very clear. This microscope, it will run for 50,000 hours. And it is environmental friendly. And other than that, when I want to see the slides on the laptop and the screen, and as well as I want to see under the microscope also, I can do both. That is 50-50 beam splitter is available in this. And other than that, if you are talking about this one, it is environmental friendly. It is having CLED also, which is directly from Euroimmun. So with this classification, we can go to hands-on. Okay. So this is the classification of the Hep2 cell pattern, which is given by international consensus on ANA pattern. International consensus on ANA pattern. It has given the classification anti-cell. AC is nothing but it is anti-cell. AC0, it is completely negative. AC1 to 14 and 29, it is nuclear, fluorescence will be there. And other than that, cytoplasmic, it will be 15 to 23, mitotic, it will be 24 to 28. So these are all the patterns we are going to see in the upcoming session. Okay. Till now, you have any doubt in this? So antibody titer is nothing but the weaker to be considered. The dilution is nothing but it starts from 1:10, 1:32, then 1:100, 1:320, then 1:1000, and 1:3200 and 10,000. This is the way the pattern are analyzed. Okay, the weaker fluorescence to be considered for antibody titer. Please remember that one. Okay, any questions? Okay. So right now we can assemble as team. Maybe this entire row can become one team, and this row can become another team, and this row can become another team. Totally 5 teams we can have, so that the mixture of the people will be there. Can we start this hands-on? Please come. The entire row. So we'll divide the people based on the row. This row, sorry, this column and this column. I don't want the same, colleague in a same batch. That's the reason I'm telling. It's working. Yeah. Santosh, you stand here. No issues. So I have allocated a separate person for each table. They will assist you in doing this test. You have a paper over it. There is a procedure paper. You can see that procedure paper. Okay. What is the first step involved in it? First step is taking the titer plate, adding the sample. The second step, then incubation. The third step again. Can you listen? Please pay attention. Please pay attention. Please pay attention. So the first step, it is written as titer plane technique. So we are adding 30 microliter of the serum on the titer plane. The second step, incubate for 30 minutes. Third step, wash it with one flush, and as well as incubate for 5 minutes with the PBS solution. Then next one, add 25 microliter of conjugate, 30 minutes incubation, flush it with the PBS solution and cool it for 5 minutes. The next step would be mount 10 microliter of the glycerol and place the slides above it. So this is the basic step. So right now, the first step, what we are going to do is, we need to add the sample to the titer plane. Okay, so this is the first step which we are going to do right now. I will show you, then you can start the procedure. So that's the first thing. The second thing, what I want to tell you, like if you are talking about the dilution, which we discussed few minutes before. So since it is a ANA profile, what is the dilution to be done? 1:100 dilution. So already what we did, we took 1 microliter of the sample and we mixed with the 100 microliter of the PBS solution. So that one already it is available in your table, on your table. You can see here. So the sample is available over your table. It is already diluted. That is the diluted sample where we can use it. Okay. Okay. So the next thing, what we are going to do, like we are having the slides on your table also. So the slides, when I ask you, you can open it and you can place it over the titer plane. Okay. So where is the titer plane? For this two table, titer plane, I have kept it here. Our team will help you to add the sample. For this two table, we have kept titer plane here. And another titer plane, it should be available here. Okay. What is the next step? For incubation, what we need PBS solution. How to prepare the PBS solution? It is a simple solution step only. Nothing but take 1 liter of distilled water, add this PBS salt, and other than that, we will be adding the twin solution also, and we'll make it as 1 liter. That 1 liter solution also we have prepared it, and we have given to you in the bottle for washing. And other than that, to keep your slides for incubation, we have kept that one also. Till now, are you clear? Till now, are you clear? We'll go the procedure one by one. Don't worry about it. So what is the first step? Take the titer plane. What is that? Titer plane. Titer plane. This is known as titer plane. Open chesara. So you can keep the titer plane over it. So what is the first step? Don't do anything. Please watch the procedure first, then you can do the procedure. Please watch the procedure first, then you can do the procedure. Okay. First of all, what we are going to do? We are going to take the titer plane in it. I am going to place it over the table. And what is the next step? The kit which has the positive control in it and as well as negative control in it. Okay, the kit which has the positive control in it and as well as negative control in it. So, the first column of the titer plane, we are going to add 25 microliter of the or 30 microliter of the positive control. Open it. Open and give it to here. We'll show it, then we can go for the next step. Madam, you have taken 30 microliter. You will have to add the 30 microliter positive control over the slide here. It will not go away from that column particularly, that cell. Give here, give here. Manoj, I will show you, then you can show it to them. Give that. Yeah, I'll show you. The first one is to be added with the, we are adding the positive control here. Please watch the procedure. Yeah. Naveen Garu, you go back side. You also can go to the back side of the team. Yeah, you can add over here. So we have added the positive control right now. Madam, discard it one. No, don't use the same. Yeah. Yeah. Please wait. Open it, Manoj. Naveen, come to this side. Come to this side. Yeah. Second column. Yeah, discard it. So in the first column of the titer plane, first cell of the titer plane, we have added the positive control. Second cell, we have added the negative control. The next step would be adding sample on the each and every cell. Okay, so I'm going to add the sample. So we're going to add the sample here. So sample is available here, you can take the sample. So this is the diluted sample. As I told you, it is 1:100 dilution for ANA. So you will have to add in the third one. Press it fully, yeah. Yeah, it is fine. Okay. So now the sample is added. Anyone else is available in this team? Okay. Now what we are going to do, we are going to place the slides over it. Just a moment. Sir, you can remove the slides. Yeah. You want to add one more sample here? Yeah, please add. So as I told you, since it is a multiplex technique, I can add up to 7+ sample. Yeah, you take that sample. Here. One minute, sir. So we have added one more sample also, second patient sample right now. Sir, remove the match. So as I told you, biochip, you should not touch over it. Okay, you should touch at the corner only. And other than that, this is the 2 biochip technology. What we are having, one is primate liver and another one is Hep2 cells. So what we are going to do right now, we are going to place this biochip over the sample, positive control and negative control. So please remember, sir, 1 minute. You have one AGU over here. You can see it on the slide. Once I ask you to open the slide, you will find it. You will be seeing one AGU over the slide. So what we are going to do right now, we are going to keep the edge over here and place the slide over it. That's it. So make sure that the solution will be absorbed by this. Then the reaction will start. Are you clear right now? So right now you can start adding the sample from the third column, positive control and negative control, we will send it to you. Then you can add the positive control and negative control. Add the sample which is available over there. And the third and fourth column. Fourth cell. Please start the procedure. No. No, sir. That is the reason we have given the edge. Manoj, ask them to add the sample first. For them, let them add the negative control and positive control. Yeah. So for you, the titer plane is available here. You can use this 2 column. For them, this is 2 column. So you can add the sample over here. 1, 3, 4, 5. You can add the sample. Please add the sample. So this titer plane, yes sir, I'm keeping it here. Since you are also available, so what we can do, you can use these 2 titer plane column for you, and as well as this 2 titer plane for this group. So you can add 3, 4, 5 samples here. So the sample is available here, 30 microliter. Then we will give the positive control and negative control to you. Raleda. I think different this one to be provided. Sorry. No, that one is not working. Yeah. Yeah. Can use this. Air bubbles should not be there. Okay, we should not have air bubbles. Sorry. Yeah. Okay, no issues. It should be available. How it will come down? So keep it over here, number 3. Don't worry, it is having hydrophilic and phobic layer. Wherever you add, it will come in the center. You will try to add there. Yes. Yeah, you can add one more sample. The same sample, you can add it. You can try it once again. Yeah. Same 30 microliter we are having. Yes. Madam, you can add the 5th one. Give the positive control, add the positive control. This is positive control. So you can add the next sample, sir. Huh? They are adding the same sample. So you can use this one also, no issues. Madam, have you kept the slide?
No.
Okay, you can open the slides. You can open the slides. Yeah. Sir, positive control you need to add. Yeah, please wait. So as I told you, the edge is available over there. Okay. 1 minute. 1 minute. See, when you are having this edge, the edge should be like this. So like, turn this way. So keep the slides and turn this way. So the edge is available. Everywhere it is available. So this is your side and keep it moving.
Manoj.
Yeah, just a moment, just a moment. Yeah, please wait 1 minute. So how to just place the slides like this. Place the slide like this, you will be having edge over it and turn it easy. Okay, so if you are placing over the edge, then it will fix it on the sample properly. Okay, fix it, no problem. Sir, you open the slides. Have you opened the slides? Yeah, it's okay. Then you can place here also positive control, no issue. You can use this as a negative control, not an issue. But discard the cells. You keep it here now. Apply it again. Apply it again on this one. Madam, you can note down the time. So what is the time? It should be half an hour from this. Please note down. Okay, those who has completed the procedure, you can have your seat, please. Madam, you can have your seat, please. You have noted down the time, madam? Yeah, please have your seat. Nothing. So, you're adding positive control and negative control. Yeah. We'll do one thing. You take it. While the meantime, we'll place the slides. Yeah. Yeah. Yes. Open the slides while the meantime. So how to place this one? Majorly, 1 minute. Okay. So you have the edge over here. You can check the edge. Okay. You can turn this side. Where is your sample? This is a sample, right?
Yes.
So place it over here, and it is fixed. Easy. So the edge should be fixed with this one. When we are talking about this edge, it should be fixed here. I will explain you once it has come. So note down the time. Note down the time. 3:30 we can come and take. Please have your seat. Please come back. Sir, please come back to your seats. We have to see almost 28 patterns right now. Please come back to your seats. Yeah, can connect. Okay. So in your bag you are having ANA pattern booklet. Can you take that kit? In your kit you are having ANA booklet with you. Huh? Yeah, PPT kaval. Okay, so I'm sorry, what we can do, we can see the machine once again and we can come back because they are also preparing this. There is one more machine. See, what we discussed initially as a first line where what diagnosis, what we need to go for indirect immunofluorescence technique. That is a gold standard. Then for confirmation, which one you need to go for? You need to go for either immunoblot or ELISA. So we are going to see immunoblot right now. Please come with me, join with me there, over there. Please get inside. Anyone is available outside? How about others? Everyone is available inside? Everyone is available? Yeah. See, when it comes to immunoblot procedure, we need the nitrocellulose membrane strips, which has the antigen coated in it. So this is the nitrocellulose membrane, which is known as optimized membrane chip. So what we are telling this as optimized membrane chip, nothing but nitrocellulose membrane are coated in a regular interval where the antigens are coated, which is also available in the immunofluorescence. Okay, through immunofluorescence technique with one substrate, what I said, we can detect up to 150 different antibodies. Okay, but when it comes to this one, we can detect, for confirmation, we can detect maximum 23 antibodies by using this one, which is known as ANA 23 profile is there. So, and other than that, we are having different profiles for the diseases. For example, if you are talking about progressive systemic sclerosis, we have a separate immunoblot profile. So, once I complete indirect immunofluorescence technique, I come to know that some of the antigens are positive. For confirmation, I'm going for the immunoblot procedure. To do the immunoblot procedure, I need this strip, which is known as optimized membrane chip, which is known as LIA, line immunoassay. So in this line immunoassay, to do the procedure, we are having the machine called PluxMat. Sir, 2 minutes, sir. Yeah. We are having the machine called PluxMat. So this PluxMat, it is having 3 variants. One is PluxMat 4, PluxMat 8 and PluxMat 16. So with this PluxMat 4, we can process 4 samples at a time. Even a single sample also you can process. With the PluxMat 8, we can process 8 sample, minimum 4 sample you can process. With the 16, which is the next version, we can process minimum 8 sample, maximum 16 sample. Okay, so this instrument is made in India instrument, which is done by our R&D of Everlife CPC Diagnostics. Majorly, if we are talking about the parts of the instrument, here we will store the reagents, including conjugates, substrate, and as well as we'll be using sample buffer also. And other than that, this is the oscillation area where the immunoblot oscillation will happen, followed by the wash buffer will be kept over here. And other than that, we'll be having distilled water over here for washing. Okay, what are all the steps will be involved in it? First one is addition of the wash buffer to this, and then the oscillation will happen. The next one is adding the sample buffer in it, then the oscillation will happen, incubation will happen for 60 minutes, then later it will add the secondary antibody to it. Then later, once the secondary antibody is completed, we'll add the substrate and close the solution with a stop solution. So it takes almost 1 hour 30 minutes to 2 hours to complete the procedure. But when it comes to allergy profile, it takes almost 3 hours 15 minutes. There are multiple profiles you can run in this machine, not only ANA profile, you can run neurology profile, you can run nephrology profile, you can run allergy profile, and C-ANCA, P-ANCA, all those things you can run in this machine itself. So right now, Manoj, so what is the samples you have taken?
You have different scripts for different profiles.
Yes, sir. It's based on the antigens only. Antigen. Yes, yes. Sir, it varies profile to profile. Maybe I'll ask my colleague to discuss with you. But for example, ANA profile, 15 antigen profile is there, 16, 17, 18.
This is around 17,100 per strip.
Sir, majorly commercial part will be taken care by my team member. So that is the reason I am not taking. Commercial part will be taken care by my team member. That is the reason. I'm including in marketing. I'm telling him the product right now. So that's what. So based on the number of antigen, the cost will vary. For example, if you are talking about 23, little costlier than the 15 antigen profile. And yeah, Manoj, over to you.
So this is the ANA profile plus DSF 70 kit. So
You continue.
So in this kit, we can get this strip rack, as well as sample buffer. This is the sample buffer. And this is the substrate. And this is the conjugate and wash buffer. So first we have to dilute the wash buffer with 1:100 ratio, like 900 ml of distilled water and 1 ml of wash buffer, this wash buffer, sir. So I already diluted the wash buffer for one test. So I am adding to the position of.
So there are two type of trays are available. One is black tray and another one is white tray. If you are talking about the black tray, the volume is little more. It is for ANA, ANCA and other autoimmunity profile. When we are talking about the black tray, which is for only for allergy profile. Okay. And one more thing, whether I can reuse the tray after the procedure, we should not reuse the tray. Then if you are reusing the procedure, the contamination will happen, the result will not be proper. So once I done the test, I need to discard the tray. Then I need to use the new tray for the next test. So some of the lab, what they are doing, they will be adding all the samples here, and after the procedure, they will wash with the distilled water and they will use the same tray. And that time blackening of the nitrocellulose membrane will happen. It will lead to false positive results. So always the tray should be used very clearly. And yeah. So we have the software which is known as PlexMat software, it comes with the system. And other than that, we have the software called the Euroline software, which also comes with the system. Okay. So Manoj, you can start. So the trays are fixed properly. Okay, use the block one right now. Yeah. So I'm going to run this test in demo mode only right now. So you are going to start user mode. Can you see the test menu over it? Kindly see the test menu over it. Please see the test menu. We can see the antigen profile 23, antigen profile 4G. We have 6G, and other than that, we have liver profile 3G, autoimmune liver diseases, myositis profile, myositis 3 profile. So these are all the different kinds of profiles are there. So for the incubation of this same, all this red color, it is same. In case if the reagent is same, means you can run at the same time. And other than that, for example, allergy profile, if you are talking, there are 4 different allergy profiles are there. These 4 allergy profile we can perform in the same time. So based on the incubation and as well as antibody, we are giving the colorization code. So you can perform multiple profiles by using this machine. So that's a major advantage of this machine. And for example, we are going to run this test. Yeah.
No, I diluted the.
You have diluted the?
No, I diluted the conjugate. 1:100 ratio.
Yeah. So the conjugate to be diluted, 1:100 ratio, he is diluting the conjugate, substrate.
Sample buffer 1350.
Yeah. Usually the test kit, it comes with the 16 pack. These are all the new strips. You can see it without touching the nitrocellulose membrane. It will be having control over it. So with this control, we will come to know indicator band. If it crosses the positive 3+, then the test is valid. If yes, and we have positive control in the solution. We have separate positive control also in this test kit itself. Okay, but with this test strip, we are having the control indicator. This control indicator, it indicates that the procedure is right or wrong. We have done the procedure rightly or not. If we have not done the procedure rightly, or if there is any problem with the sample, then the positivity will not be there. It will be in between either single plus or borderline plus. So it should cross plus, 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 3+. Strong positive, I can say. Okay. Sir, how many of you are using this LIA, line immunoassay, in a rock shaker, or are you using LIA? How many of you are using LIA, line immunoassay? Blot. TB you are using. Okay. Okay. Fine, fine. So maybe okay, sir. Fine, sir. Autoimmunity. Okay, 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 sir. Probably when you are starting this procedure, in case, for your lab or the known lab, when you want to start this procedure, it's better to start with the manual process. For example, you will be having rock shaker. The simple rock shaker would be fine to run the test when you are receiving 1 or 2 sample in a month or in a week, and that time start with a simple rock shaker. So whatever the test kit information which is given, we need to add manually. For example, when I want to add the sample, we need to add the sample manually. When I want to add the conjugate, we need to add the conjugate manually, substrate manually. All these things, when you have the lesser workload, you can start manually. But when you want to go for you have the better number with you. For example, you are having around 5 or 6 or 10 samples in a week, then go for the machine. So that will easy your process. Okay. So we are not selling the machine. We are not here to sell the machine. We are selling the concepts only. Okay. So you decide based on your convenience, based on your lab, the number of samples which is available in the lab. Start with the manual one. When we are promoting this product continuously, we will increase the workload, then go for the automation, which will not affect your ROI basically. So that's the main thing which I want to convey. Manoj, you can tell what are all the things you have done. Right now he's placing the new step, which is nothing but ANA DFS. ANA DFS. Yeah. So what are the sample dilution you have done? You can explain.
Okay. So I did a sample dilution with 1:100 ratio. So I'll take in 1500 microliters of sample buffer, red cap. Yes, ma'am.
So requesting you to pay your attention, please pay your attention.
So now I am doing the sample incubation. So meantime, I will do the start the test. So I'll take in substrate around 1500 microliters. So this is the position for substrate. And I diluted the conjugate into 1:100 ratio, like 135 of sample buffer and 150 of conjugate. I dilute it into 1:100 ratio. After that, I'll add substrate 1500 microliters directly to this jar. And for strip activation, we need sample buffer. So already I placed 1500 microliters here. So I already placed that strip inside the rack. This is the strip position. So I placed the strip here. And the wash buffer already prepared. And which one, sir? Sir, ఇది వచ్చేసి శాంపుల్ బఫర్ 1500 మైక్రోలీటర్స్ కిట్లో ఉంటుంది. ఇది వచ్చేసి కిట్లో వచ్చేసి శాంపుల్ బఫర్ ఉంటుంది, సబ్స్ట్రేట్, ఇది పాజిటివ్ కంట్రోల్, ఇది కాంజుగేట్, ఇది వాష్ బఫర్. సో నేను శాంపుల్ బఫర్ తీసుకున్నాను.
దీనికి ఏం లేబుల్ లేదు.
లేదు,
ఓకే, మీరు దాంట్లో
శాంపుల్ బఫర్ తీసుకున్నాను. సో ఇది వచ్చేసి సబ్స్ట్రేట్
సబ్స్ట్రేట్కి ఇంకొక సపరేట్
ట్యూబ్ ఉంది. అది వచ్చేసి ఇది, అది 1500 మైక్రోలీటర్స్ డైరెక్ట్. 1500. ఇది కాంజుగేట్ వచ్చేసి శాంపుల్ బఫర్ విత్ కాంజుగేట్ తోని మనం డైల్యూట్ చేసుకుంటాం.
ఇప్పుడు క్లారిటీ వచ్చింది. ఓకే, తర్వాత యాక్టివేట్ చేయడానికి స్ట్రిప్ యాక్టివేట్ చేయడానికి ఏం యాడ్ చేయాలన్నారు?
అదే శాంపుల్ బఫర్ ఆటోమేటిక్గా మెషిన్ పెట్టుకుంటాం.
You can see this already we have kept the strips over. Place the strips here. Start the position. Now already we have placed the adapter, and other than that we have kept this tray also. Okay, and as well as place the strips also. He has selected the ANA profile right now, 3 here, and the color has become blue color. You can see that. Yes, he has selected that. So now he is starting this test. So there are 2 modes. One is demo mode and regular mode. Right now we are running in regular mode. Regular mode we are running. So enough volumes or samples are already kept here. Conjugate, substrate, and as well as wash buffer we have kept over there, and he is starting the procedure right now. Then the rocking will happen right now. That you are having that sample addition, right? As a first step, it is taking the sample buffer. Yeah. So first step, it is taking the sample buffer for the incubation. So it will activate the antigen basically. So it is activating the antigens right now. Sample buffer 1.5 ml. It takes at least 2 to 3 minutes to activate that. The shaking will happen right now. After that, the samples to be added. Once we add the sample, then we can go to the next procedure. Sir, if you are having higher number of workload, for example, if you are having 40 or 20 plus samples in a day, then we are having the separate instrument for that, which is known as Euroblot 1. So those Euroblot 1, it reads a LIS, it reads a patient sample and enters everything in it. So you'll be having number on the strips. So you can see the timer also here. It takes 3 minutes to complete the entire incubation. Maybe it takes 5 minutes, 2 minutes are completed right now. We have 3 more minutes to complete the incubation. Once the incubation is completed, then we will add the sample. So the sample addition will be done by manually. Okay, what we can do, the first test what we kept, it is completed the half an hour timing. So we will wash that and then we can come back and see that. Okay, till that you can hold it. Yeah, we can go there and we can do the test. Yeah, please come. See, come and assemble as a group. Come to your table. Please come to your table. Please come to your table. Please come to your table. Okay. See, for this slides, the incubation is completed for 30 minutes. Maybe for you it is 3:30. I think for you it is 3:27 or something. So what we can do right now, I'll show you the procedure. Santosh, please come. Hold this mic. So I'm going to remove the slides. I have removed the slides. What I need to do, I need to hold the slides like this. I need to hold the slide like this. Give that wash buffer, this PBS solution. That PBS solution, sorry, this one, no issues. See, the blue color lid, you are having the PBS solution. Okay. So what I need to do, I need to flush it once only. Madam, you open this. Okay. What I need to do, I'm going to flush it once. Once it is completed, then I will soak it PBS solution for 5 minutes. Close it. It's okay. Just you can leave them. So it is 3:25. So whoever have completed that, splash the water. Please don't pour the water again and again. Only one time. Only one time. So you have PBS solution in this bottle. Please pay attention. Please pay attention. You have PBS solution in the bottle, which is in front of you. For washing, you can use this PBS solution. Yeah. Madam, do one thing. Take this slide, keep it inside. We will be adding the conjugate right now, which is light sensitive. So this for you. Take the slide. Yeah. Hold it. Okay. He also this also to be washed. No issues. Nothing is So have you washed it? Completed the procedure this side, madam? It is done. 5 minutes. Do one thing. You can. Okay. That when. What is the timing for that, madam? Okay. Yours is the last one. I think so. Madam, do one thing. Take the tighter plane. Get inside the room. Keep it inside the room. We are going to add the conjugate. Don't close it. Don't close it. Just take the tighter plane and keep it over there. Inside the room. Only once. Not twice or thrice. Okay, flush it. Flush it. Flush it. Again. Pour little. Yeah. If antibody goes means then don't ask me where is the antibody. Don't search the antibody over there. Don't close it tightly. Just put a lead. Put a lead. No issues. Okay. How about here? 3 minutes. Huh? 3 more minutes. Okay. You can come with me. Okay. Just a moment. Just a moment. These 2 batches, 3 batches come along with me. We will add the conjugate. Bring your pipette and tips also. You cover this one. Please come here. Please get inside. I am not going to give microscope now. Please wait, please wait, please wait, please wait. Don't switch on the light. Okay. So this is yours and that is them. So this is the conjugate solution. This is the, please see here, this is light sensitive. That is the reason I have asked you to come here. So where you have done the test, this is the first column. Then second column, 1, 2, 3, 4, 5, 25 microliters of the conjugate to be added. Adjust into 25 microliter. Okay. Sir, can you open this? Yeah, same amount, same amount. Please continue with the same amount. Please wait. All the 5 columns you need to add. Five cells you need to add. 1 minute. Yeah, please tell me, madam.
You should add here, huh?
Yeah, you should add here.
Not this one.
Not this one. So you are going to completely change it, keeping the slides over it. Open the door, no issues. Yeah. Add the conjugate again. Door open cheyandi. Bas. Wait here. You keep it like this. Hold it here.
Watch it.
Keep it inside.
5 minutes.
Kept it? Yeah, completed? Finished, na? Take that. Before that, take the tissue paper. Take the tissue paper.
Tissue paper here. Take. ఏదైనా పర్లేదు.
Hmm?
Take.
Take this slide to there, otherwise it's in background.
Yeah.
So we do trying here.
What? Conjugate you are asking, na? Conjugate we are adding there only.
No sir, slide will take there.
Hmm. Come.
You bring your slides.
Okay, have you added? Is it completed? All are completed? Okay. Madam, can you come to this side, please? Yeah. So, do one thing. Can you hold this mic? Madam. 1 minute. So what we are going to do, please remember that we are going to add the conjugate right now. One minute, please hold. Okay. See, we have taken the slides back. We have taken the slides out right now. Wipe it on the edges only. Wipe it on the edges needed back side. Don't wipe it on the other sides. Okay. Now, then what we are going to give? We are going to keep it over the conjugate here. Sorry. Just one minute.
That is not the first slide, no?
మేడం మాదే కాదు మేడం మాదే మాదే
You wipe it up. Sorry, wipe it up. Can you give me tissue paper? Give that slide. Have you added all the conjugate?
5 members.
Add one more.
హలో వినిపిస్తుందా నారాయణ?
Additional volume of conjugate was added. Maybe instead of pressing once, you have pressed it twice. Instead of pressing once, you have pressed it twice. The conjugate has completed right now. No, not you. I'm not telling you. Maybe the conjugate when you are adding this one, we have pressed it twice. Okay, fine. Not an issue. Give the other slide. You have conjugate over it here. Sorry, madam. Stay here. Just a moment.
అందులో మనం కంట్రోల్
వేసేద్దామని చెప్తాను
థర్డ్ నుండి స్టార్ట్ అయింది
Okay. Okay, so we should add it in the second one. Please wait. Okay, anyone is having the slides with you right now?
yes.
Yeah, I'll take it.
ఫస్ట్ యాడ్ చేసిన వాళ్ళు కొద్దిగా వస్తాయి 5 members యాడ్ చేసి
So whoever has
Very-
completed that, you can leave. Maybe that slide, it is little complicated only. Well, let's manage, don't worry.
You are adjusting same 25 microliters over to
Sorry, madam.
same 25 microliters you are adjusting for two sides now.
No, I'm not reducing it. I'm taking it. While pressing itself, it is coming with additional volume only. I'm adding enough conjugate, don't worry. Yeah. Okay, right now,
This is a
how many of you have kept the slides? Can you give me some tissue paper? Tissue paper.
టిష్యూ పేపర్ టిష్యూ పేపర్
Hmm. Sorry?
అదే టిష్యూ పేపర్ కావాలి
Yeah, thank you. As I told you, please remember. Okay, you want to speak it in mic. Please remember that. చూడండి ఇక్కడ చూడండి. Sir, please. The wiping should be on the edges. Don't touch the biochip. You can see here. The edges should be wiped off. And if needed, if you are confident, the back side of the slide can be wiped off. Are you clear?
How you know, sir, which side is there? That
See, this edge is available here. You can see here, the edge is available over here. So, I am going to place this strip over here. Now you can see the difference. Now we can see the difference, how it is fixed over there. Okay? Sorry? Yeah, we should not have bubbles in it. It will affect it. Right now, the some additional samples are added. That is the reason I am managing this. Okay? Don't worry. Things will be all right. Okay, so this one it is done, na? Slide is there, na? As I told you, always check the edge, where the edges are available. Okay, the edge is available here. Again, back side, this one, and this one, and place it over there. Yeah, edges. Actually, this should be in this way. We should not have conjugate on the outside. Okay. So now we have kept two slides, and where is a box? Can you give me some that tighter plain box? Yeah. So what I'm going to do? I'm going to close it with this one, since it is a light sensitive one. Okay. Please wait. And one more strip has to come. You are having the one more strip, sir. Next one. Yeah.
One slide is in the code.
Sorry, one slide is.
In the code.
Yeah, we'll take it. So whoever have completed the Kuwait, then they can take the Kuwait view. This one, I think this one is completed. Yeah, this one also it is completed.
Or did you?
Sorry. Can you bring some notebooks? Notebook. Five notebooks. Some notebooks.
5 notebooks.
Ah,
5
Yes, sir. So where is the other Kuwait? So it is available here.
నెక్స్ట్ ఇక్కడ 30 మినిట్స్ బై సెంటర్ క్లాస్ అవుతుంది ఇంత క్లాస్ చెప్పాను అక్కడ అవుతుంది 30 మినిట్స్ క్లాస్ అయినా అవుతా ఇవ్వచ్చు లేదంటే మిషన్ దగ్గరికి వెళ్ళచ్చు రెండిట్ లేదంటే
So two things are happening. Just for that. No, light sensitivity, we have kept this one. And one more slide is there, right? Yeah. So where is that? Yeah. In that, yeah.
Notebooks tension.
Yeah, where is that, sir?
Here.
Then smaller size will be fine, sir. Just for complete this one, light sensitive. To make it light sensitive. One minute. Please.
Smaller size.
Smaller size.
Wait.
Okay, there is a slide, another slide, it is available in this map.
Sir, I have a doubt, sir. We have added the positive control, negative control, and samples in this.
Yes, sir.
You chose another row to add the conjugate. You chose another row to add the conjugate.
Yes.
Not the ones which we added.
Yeah.
Okay.
Because it is a secondary one. Sorry, sir? One slide, sir. I didn't get you.
From this side.
On this side? And bring one only one notebook. Give me one notebook.
In the book, sir. It's in the sir.
Yeah, like that you can keep. Sufficient. Done. Okay, please come. Bring this one.
Next.
Pattern. Team, please understand. Please get back to your seat. Kindly get back to your seat. You have added the sample, huh? Add the sample. Kindly get back to your seats. Kindly get back to your seats. Okay. Right now the time is 3:45. By 4:15, 4:10 we can go for the, next step washing and addition of glycerol. And we can see it under the microscope. So connect this system. Okay, a quick recap. Sir, please. Okay. Just we'll start the, whatever we have done the steps. I want to have a quick recap. So initially what we did, we took the titer plan. So with this titer plan, what we have added the patient sample, positive control, negative control, followed by patient sample. So after that, what we did, we took the slide, we kept over it, we incubated for 30 minutes. So it was completed. Again, we took that one, we washed it, and we kept in the PBS solution for 5 minutes. Then we removed that, and we have added the conjugate right now. Then the incubation is for 30 minutes. Still, as of now, we have completed this one. The next step would be washing that slides and adding the glycerol. That is PBS solution 5 minutes. Then later we'll add the glycerol, and we can see it under the microscope. Okay. As of now, you have any doubt in the procedure? Sir was asking like, I'm adding the sample in one column, and when I want to add the conjugate, I'm adding it in the second one, because you will not be having time to wash that titer plane and dry the titer plane. So that is the reason always use 2 column to wash and as well as adding the conjugate. So per slide test, use 2 columns of the titer plane. Okay, so that is the reason we are using that one. Okay, so we are coming to the most interesting, the pattern one. So when it comes to this one, fluorescence, it plays a major role. It plays a major role in indirect immunofluorescence technique. As I told you the classification earlier, you can see the classification. Yeah, 1 minute. So this is the classification we have seen. What are all the classification we have seen? Negative, which is anti-cell 0, nuclear 1, AC1, that is anti-cell 1 to 14 and 29, and cytoplasmic, which is 15 to 23, AC anti-cell, and other than that, mitotic, which is given by international consensus on ANA pattern. So this is the pattern which we have given. It was given by ANA. So we are this classification, if you are seeing that nucleus pattern, which is coming to the fluorescence on the homogeneous part of the nucleus, and nuclear membrane, only the membrane part will be having the fluorescence, and other than that, nucleus granular, there will be granular pattern on the fluorescence on the nucleus. And other than that, dotted, the number of dot pattern will be available. Fluorescence dots will be available on the nuclear pattern. And other than that, nuclear will be available. If you are talking about cytoplasmic pattern, cytoplasmic granular pattern, cytoplasmic fine granular, and cytoplasmic filamentous, all these patterns are coming under the classification. We need not to remember right now this thing. Let us see the patterns one by one. coming to the pattern number 1, which is known as AC1. If you want, you can make a note on this. And other than that, in that book also it is available. Majorly, the AC1 anti-cell 1, the entire nucleus will be having the homogeneous pattern. The fluorescence will be seen on the entire nucleus. When it comes to the part, which are all the antigens are targeted in it. If a patient is having SLE, the antigens like dsDNA, ssDNA, nucleosome and histone, everything will be targeted over it. If the fluorescence appears on this, then the disease is very clear. It is SLE, drug-induced SLE, and as well as juvenile idiopathic arthritis. So these are all the conditions when you have nuclear homogeneous patterns which you have. So the fluorescence on the entire nucleus we can see. You can identify by using this pattern. And other than that, if you are talking about the primate liver you will be having coarse to fine clumsy fluorescence on the nuclei. So this is the first pattern which says for SLE patient the homogeneous fluorescence will be available on the entire cell nucleus. This is the AC1. I am going to the next one. A patient, I want to identify SLE very specifically through the fluorescence technique. You have the organism called the hemoflagellate called Crithidia lucile, which is known as Crithidia lucile. It is having gained mitochondria in it. It is having gained mitochondria in it. When you are using this fluorescence testing, that is indirect immunofluorescence technique, the fluorescence will appear on the kinetoplast. Both kinetoplast, which is on the antibodies, will be appear on the both the kinetoplasts we can identify. So what is the indication? The major indication will be on the SLE. But when it comes to this one, antibody against the cell nucleus, if we are having one fluorescence in the cell, either in the basal body or either in the cell nucleus, then it is completely negative. So you will be having a separate cell, Crithidia lucide, to find out the dsDNA, double standard DNA, which indicates systemic lupus erythematosus. So till now, we have seen only one anti-cell classification, which is nuclear homogeneous followed by dsDNA with the Crithidia lucide. Okay, I'm coming to the next one. So I'm taking the cell, I'm taking the ANA, and as well as I'm seeing the ANA IF under this pattern, I'm doing the procedure. When I get the dense fine speckled pattern, which is having uniformly distributed dense filed speckled patterns on the nucleus. So we can see that dense fine speckled. So what is the difference between the previous one? The previous one, you can see that the immunofluorescence was available on the entire nucleus. But here the fluorescence are dense fine speckled fluorescence, which indicates atopic dermatitis, asthma, and interstitial cystitis, which is coming under AC2. I'm going to the next one. What is this? This is coming under centromere protein. So this pattern we can identify very easily because how many dots are available? The dots will be available. How many dots are available in this? We can say it is having 46 to 92 dots are available in this. So when you have multiple number of dots in the nuclear pattern, 46 to 92 dots, which is indicating the centromere protein A and B, which is indicating the centromere protein A and B, the indication is very simple, progressive systemic sclerosis. So most of the progressive systemic sclerosis cases, if you have the nuclear dots, which is 46 to 92, then you can confirm with this one, then go for the immunoblot procedure. Okay. I'm coming to the next one. If you are talking about the next one, that it is against the nucleoplasm. You can see the nuclei, many of the nuclei are not having fluorescence, only nucleoplasm is having the fluorescence. So which indicates very clearly it is targeting SSA and SSB, which is nothing but Sjogren syndrome. Sjogren syndrome A and Sjogren syndrome B antigen it is targeting, the antibodies are producing against SSA and SSB, which is indicating Sjogren syndrome. What is the pattern? The pattern number is AC4. Okay. we have a quick recap again. As of now, we have seen 4 patterns. AC1, AC2, AC3, AC4 also. AC1, nuclear homogeneous. The entire homogeneous was having the fluorescence. It is indicated for SLE. It is indicated for SLE. The next pattern what we have seen Crithidia lucidae. So in this Crithidia lucidae, what we have kinetoplast and as well as basal body, both are having fluorescence. I'll repeat again, you can see here. So both the antibodies are having, both the in dsDNA, in cell nucleus and as well as basal antibody, both are having fluorescence, which is pertaining to SLE, systemic lupus erythematosus. I'm coming to the next one here, dense fine speckled, which is for atopic dermatitis cases. The fourth one, coming to AC3 pattern, centromere A and B, 46 to 92 dots will be there, which is targeted for progressive systemic sclerosis cases. The next pattern, if you are talking about the next pattern is for nucleoplasm, which is targeting on Sjogren syndrome A and B antibody, where the fine speckled fluorescence of the cell nuclei in interface cell and as well as it is slowly silhouetted from the nucleoplasm also. You can see it is slightly silhouetted from the nucleoplasm also. We can identify the fluorescence. Based on the fluorescence, you can identify the diseases also. So majorly it is targeted for Sjogren syndrome. Yeah. I'm going to the next pattern, which is AC4, anti-cell antibody, anti-cell pattern 4. So in this, it is targeted again Ku protein, which is targeted for, this is the targeted antibody for dermatomyositis. So when we are talking about Mi antibody, when we are talking about Ku antibody, which is mainly related to dermatomyositis and polymyositis. So these are all the conditions it is targeted. How to identify that? Majorly we are having large fine speckled fluorescence of the cell nuclei, and other than that nucleoli also we will be having. But when it comes to primate liver, clumsy speckled staining of the nuclei we can identify. When it comes to Ku antibody, always refer the primate liver pattern. When we are referring the primate liver pattern, so you know the clumsy speckled cell nuclei we can identify very easily, and which is targeted for Ku antibody, which is polymyositis and as well as dermatomyositis cases, which is visible. And the next one, Mi2, which is also coming under AC4 pattern only. Here, it is also coming under fine speckled fluorescence pattern, and the disease is dermatomyositis. As I discussed with you, if we are talking about Mi2 antibody and Ku antibody, if the fluorescence is available on the nucleus and nucleoli also, it is targeting for dermatomyositis and as well as polymyositis. Okay, so this is the next one. It looks like in primate liver, this antibody, what is the antibody we are targeting? We are targeting U1 RNP SM RNA polymerase. It looks like a cotton candy in the primate liver. In the primate liver, if the antibody or the fluorescence, if it is having the cotton candy-like structure, then it is targeting HN RNP, UN RNP antigens, and the disease is mainly SLE and as well as multiple connective tissue disorder. So as of now, we have seen 4 patterns. Please remember that these patterns we will have to see again and again. When we are seeing the fluorescence again and again, we will come to know reach the expert level. Right now, since it is the workshop plus this one, I'm showing the fluorescence and indications only. Don't think that we can identify everything right now. It will take time. Little time in identifying this one. As you know, many of you know this. Okay. So earlier we identified one centromere A and centromere B. So the nuclear dots were 46 to 92. In the similar way, the nuclear dots, if it is having 6 to 20, means it is targeting SUMO antigen. It is targeting SUMO. We can count the number of nuclear dots 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7, 8. So the dots are from 6 to 20 nuclear dots will be in the this one. I'm sorry, cell nucleus, and the number of dots will be little lesser when compared to Hep2 cell. Majorly it is targeted for primary biliary cholangitis patient. Okay, I'm coming to the fewer dots pattern. Nuclear fewer dots, which is coming under AC7, which is focused where we will be having the dot numbers are 1, 2, 3, 1, 2, 3, 4. We will be having 6 dots in a single nucleus, so which indicates the Sjogren syndrome. The target antigens are P80 coilin and other than that SMN also. Okay, I'm coming to the next one. See, earlier patterns, when we identified those patterns, those patterns, either the nucleus, all the nuclei will be fluorescence, or you will be having fine speckled, or you will be having dense fine speckled. But when it comes to this one, you can see the homogeneous fluorescence on the nuclei, fine speckled reaction to the nucleoplasm. We can see the fine speckled, fluorescence on the nucleoplasm, which comes under AC8, which is the antibody is PMSCL, which is focused for it is polymyositis and as well as systemic sclerosis patient. It is targeted. So, how we can show the difference? You can see the difference with the dots. You can see the difference with the dots. And other than that, fine speckled pattern. This is the fine speckled pattern. But when it comes to this PMSCL, you are having fluorescence or the speckled reaction over the nucleoplasm also, which is focused for PMSCL antibody, progressive systemic sclerosis, overlap syndrome will be there, and polymyositis will be there. I'm going to the next one, which is AC9. When it comes to AC9, you can identify very easily the fluorescence on this one, which says the mitotic coronary perichromosomal fluorescence will be there. Mitotic coronary perichromosomal proteins will be there. Fluorescence will be there, which is targeting U3-RNP fibrillary, and the disease associated with the progressive systemic sclerosis. The progressive systemic sclerosis. Okay. See, when we see the pattern continuously one by one, we can identify the disease very easily, but we will have to see the multiple patterns. And this is one. It is known as RNA polymerase, which is coming under AC9, anti-cell 9 classification. In that, the tiger paw-like structure will be there, the fluorescence, granular fluorescence on the nuclei, and fine granular smooth fluorescence on the nuclei will be there. So, which is focused for progressive systemic sclerosis. But whether I can identify with this primate liver, I need to identify with the Hep2 cells only. So, the previous pattern, if you see, the main thing which we took is perichromosomal fluorescence, and other than that, speckled fluorescence on the nuclei to identify U3-RNP for progressive systemic sclerosis. But the next one, I am using the fine speckled, it look like a tiger paw, which is associated for the RNA polymerase for progressive systemic sclerosis. So, these antibodies, when you are identifying in this way, we can have the differentiation in this, and for confirmation we can go for immunoblot. So, I am coming to the next pattern, which is AC10 NOR90 antibodies, which is targeted for NOR90, and it will be having only few little fluorescence on the condensed chromosome materials. And if you are talking about that, the few little dots we can identify here also. We can identify here, and other than that, speckled fluorescence also we can identify here. Again, it is progressive systemic sclerosis. So, whatever we have seen for progressive systemic sclerosis, NOR90 antibody, if you are talking about RNA polymerase, if you are talking about U3-RNP, so these are all the same indication which is having. I'm going to the next one, nuclear membrane. Okay, how we can identify? only the fluorescence sun seen on the membrane of the nucleus, and we can easily appreciate the primate liver. The membrane will look like a onion rings. Okay, what are all the target antigens? Mainly lamin A, lamin B, lamin C, receptor GP210, which is focused for primary biliary cholangitis. Okay. As of now, any doubt is there? You're running very fast. Okay. See, what I am trying to say, like this fluorescence, when we are seeing the pattern again and again, okay, the different kind of mixed patterns also you will be receiving in the future. So on those times when you are comparing the patterns, you know, you will be at the expert level to review the same. Okay. Even there was an experienced, the microbiologist who was reviewing this data, and he was telling that a person who starts reporting this pattern, even for 4 years, after 4 years, their expert level will be 70% only. So when we are continuously doing this, we'll come out of this and we can report in a better way, don't worry. But the main thing is that the basic thing, once we understand that, we can report in an easy way. Coming to the next one, the antigens against PCNA. The cell cycle dependent fluorescence pattern will be there. Interphase cells exhibit the bright and fine speckled fluorescence. I can say here we can see the fine speckled fluorescence over it, and primate cells it will be largely negative, and the disease association is SLE. Okay, the 14th pattern, and we have one more pattern in the nuclei pattern. Majorly homogeneous nucleus pattern. Majorly, it is targeting centromere protein F. What is the main thing? Staining intensity varies strongly from G2 phase, and we can say pro-meta and meta phase. Small matte aligned dots are available, and nuclear membrane often slightly stained. So we can see here the cell division has happened. The fluorescence we can see over this, and other than that, this metaphase also, the two fluorescence we can identify by using this immunofluorescence. Majorly the antigen associated is CNP, centromere protein F, and this condition, malignancy, but there is no clear picture on this. This is the, I can say we divided this pattern into nucleus pattern, we divided this pattern into cytoplasmic pattern and mitosis pattern. So among this is the last pattern in nucleus pattern. So this is known as SCL70 top isomerase 1 pattern. Here nucleoli are positive, mitotic cells, the border area of condensed chromosomes are fluorescence. You can see the border area of the condensed chromosomes are positive. Here also we can identify, we can appreciate this one also. And other than that, the homogeneous fluorescence of the cell nuclei, which is seen in the primate liver, it looks like a rava laddu. That's the reason the picture was given over there. Okay, so association is progressive systemic sclerosis. Okay, as of now, we have seen this 14 pattern plus one pattern. There is 15 patterns also, which is available in your ANA booklet also. You can go through that once you, get back to the room. Right now, the time is 4:10. It's time for the incubation is completed. We can go for the washing step. Yeah, please. Please bring your, this PBS solution towards that one. Please bring the coupling jar, it is available here. And sir, give that beaker. Yeah. I don't know எப்படி வரப்போகுதுன்னு தெரியல பார்ப்போம். Let's see. Okay. So what I am going to do right now, I'm going to remove this. I'm going to wash this. Once it is done. So that is the next step. So next solution, we are taking this. I'm washing this once, flushing out, then keep it inside that PBS solution. Okay, we can use the same thing for also this one also, no issues. At a time 3-4 slides can kept inside also, not to worry. The coupling jar is for 10 slides. Close it. Yes, madam. Conjugate is a secondary antibody. It will react with that one. Can open any one of this. We'll wait for 5 more minutes. Come, please. We have processed your slides also, whoever not seen that. Okay, come back to the session. I know that it is little hectic. We are moving here and there. We are showing this procedure and we are going to immunoblot procedure. In between, we are showing the pattern also. Okay, so for time constraint, we are running in this way. We are sorry for that. And as of now, we have seen this AC pattern, AC 1 to 14. Sir, AC 1 to 14, and as well as we have seen the top isomerase pattern also, which is coming under nucleus pattern. So which is completed. Now we are going to see the cytoplasmic pattern. So this is the cytoplasmic pattern. So the fluorescence will be available on the cytoplasmic area of the cell. Okay. So what is this? F actin. This is a fiber. the fluorescence pattern using the cell line VSM, vascular smooth muscle 47, several bunched fiber structures on the fluorescence, and we can see the bile duct caniculae also in this. So what is the target antigen for this VSM? This one, it is F-actin. Majorly it is targeting liver diseases like autoimmune hepatitis, alcohol-induced hepatitis, and other than that, primary biliary cholangitis also. So when we think about F-actin, the fluorescence, if we are appreciating the fibers, by using the fiber, and we are seeing the caliculi and this bile canal caliculi and the primate liver, then we can appreciate that it is mainly for F-actin. So majorly for autoimmune hepatitis, primary biliary cholangitis, and other than that, alcohol-induced hepatitis also, it will show in this pattern. So as I told you, this is the cytoplasmic pattern, which is 16. The previous one was 15. You can note down, this is the 15th one, this is the 16th one, which is targeting vimentin. So but this vimentin, the unclear or rare antibodies are there. We are not focusing on this pattern much, but there is no clear indication for this vimentin. But the pattern will be cytoplasmic pattern. So I'm going to the next pattern. So in this next pattern, it is vinculin. So if you are talking about this vinculin, there is the increased staining of the short section regularly spread along the stress fibers of the cytoskeleton, which we can see. The basal boundary surface of the endothelium stroma and sinusoids, glomeruli, which is in the kidney, mainly glomeruli, tubuli, which is showing fluorescence here. So it is, we can see the cytoplasmic fluorescence on the cytoplasm, which is we can say dense, fine. And other than that, the targeting antibody for this only will be myasthenia gravis, ulcerative colitis, and as well as Crohn's diseases. But not only with this Hep2 cells, we can identify by using the rat kidney also. In this rat kidney, we can see the glomeruli and tubuli, which shows a fluorescence. Glomeruli and as well as tubuli, which shows a fluorescence. By seeing this fluorescence, we can identify the, Crohn's disease, ulcerative colitis, and as well as myasthenia gravis easily. The pattern comes under AC17, anti-cell 17. Going to the next one, antibodies against lysosome, which is easy to identify, very easy to identify. So among these patterns, which pattern is easy to identify for you? Nuclear darts will be there definitely. Okay. Sorry, I didn't get you. Homogeneous. That's the first one. It is very easy to identify. Sorry, sir? Okay. So nuclear data, there are three patterns, 46 to 92, 6 to 20, and we are having 2 to 6. Cotton candy. So that one we can easily identify. The similar way, if we are talking about this, it looks like a rava dosa, spreaded rava dosa. You can see the hotel, if you see, okay, like you will be having the lysosome GWB protein. The fluorescence will be seen on the multiple dots. It will be having multiple dots. The fluorescence will be look like a lysosome. Fine to medium coarse droplet shaped fluorescence will be cytoplasm. Okay, so what is the main disease which is focused for it? It is mainly primary biliary cholangitis, neurological diseases. Sometimes with the healthy individual also they will be having this ravadosa. Okay, but to identify clinically and as well as primate liver also we can compare, which is coming under AC18. Okay. So now the 5 minutes is completed. So what we can do? We are going for the mounting. Okay. Please assemble here. I'll bring the, give the, what to do? Cover slip to you. We can apply the glycerin and we can see the, slides. Then later we can cover the rest of the patterns. Cover slips. Petnada. So under that one. What should be available there? Take that. It's a glycerol bottle. It was available in this. Glycerol bottle. Okay. Cover slip ichada. E&D. Give one cover slip here. So this cover slip, what am I going to do? I am going to place it above the sheet, this thermocol. You can see it here in the first column or third column, wherever you want, you can place it over it. Yeah, please. So the number is visible to you 1, 2, 3, 4, 5. So in 1, 2, 3, 4, 5, you apply the small drop of glycerol, sorry. Very, very minute drop. It should be 10 microliter. Don't add too much. Don't add additional quantities also. Add little amount. Within that one, number 1 and 2, 3, 4, 5. All 5 dots should be there. Manoj? Again, I'm telling you, please don't touch the biochip. Use this one and this one. And slowly wipe it on the back side. Okay. Yeah. So it should be wiped on the back side.
How do you know the back side and front side?
So you have a column here. You can identify the column here. Okay. So the column, it should be on the left side. Okay. So then you can place it. Now it is taken. So it is applied to it. Please wait a minute. So I'm taking the second slide. I'm having the, you can see the edge over here. I'm wiping off it on the back side. Okay, on this side and this side also.
ఎందుకు
స్లైడ్ సరిగ్గా సెట్
వేర్ ఇస్ ద గ్లేజరా వేర్ ఇస్ ద గ్లేజరా
It is available here.
ఓకే దట్స్
So the cover slip is sticked on it. Okay, I'm keeping the second slide. Please wait. Yeah, this is the first one. Who wants to do the third one? Please come. Please come, sir. Please come, sir. Please come, sir. You can do the next one. You can do the next one, sir. No issues. Okay. So, so you keep it here. Hold it like this. Left side is there. Wipe it on the above side only and the edges, and remove the water. Okay, I'll give the.
ಸರ್ ಇದು ಟಾಪ್ ಲೇಯರ್ ಇರ್ತದೆ ಟಾಪ್ ಲೇಯರ್ ಇರ್ತದೆ
So this edges on this side. This is the top layer. So left side this one. Okay. Only edges. And place it above the. Just remove it. Water once again.
ఓకే చెస్ట్ పెట్టాలి
1 2 3 4
ఇలా పెట్టి వచ్చి
Remove the water. Central water. Leave it. It is fixed.
స్లైడ్ పెట్టి
అలాగే లేదు సార్ కట్టండి సార్
ఇది ఇక్కడ కదా
ఇది ఒకటి కదా సార్ ఇది
హెచ్ఫోన్ తీసి పెట్టేద్దాం
పెట్టండి సార్ మీరు
సిక్స్ రూపాయలు తీసి
ఇంకోటి ఉంది కదా సార్ ఎవరితో ఒకటి
దీంట్లో మీకు
పెట్టేయండి సార్ పర్లేదు చూడండి సార్
చేస్తారా లేదు అది కాదు సరే
అదే సార్ అంతే
ఈ పైన క్లీన్ చేయాలి
పైన క్లీన్ చేయాలి
పైన
పైన
అలా పెట్టేసారు టాస్ రిపేరింగ్ ఇటేసారు సార్ ఇటువైపు ఇటువైపు
చూడండి 1 2 3 ఎట్ కనిపిస్తుంది అది వీటిలో దాని మీద
దాని మీద ఇది పెట్టారు
అంతే ఇలా పెట్టి
వచ్చేస్తుంది కవర్స్ వండుకోవడానికి కవర్స్ పట్టుకెళ్తారులే చూస్తారు డైరెక్ట్ మైక్రోస్కోప్ ఇట్లా చూడాలి సార్కి
ఇచ్చారు సర్వీస్ పిజ్జా
Excuse me, please excuse me. Sorry, excuse me. Madam, please. See, so this is the slide where we have processed. During the process, this is one among your slide. I'm not dividing this into T because the conjugate, additional conjugate has gone in between. So we adjusted that one. So we selected this slide. This slide, it is the positive control one. The first column, it is showing the positive control. Totally 5 things we have done. Okay. So among the 5 one, positive control, negative control. After that, few positive samples we did. So when we did this one, we can see this is the positive control of nuclear homogeneous pattern. So this pattern has come with the fluorescence of the nucleus, which you can see. I will show it on the screen also, don't worry. Right now, for time being, I am showing it in the laptop. I want to take you as a batch. For time being, I am showing as a this first one. Manoj, move the next one. You can see that. Can you see that? It's gone, huh? It is 40x, sir. Switched on. Yeah. As I told you, in the earlier also I discussed you this microscope. When you want to switch on the microscope, immediately it will switch on. You need not wait for some time. Okay. Since you can see that first one, Manoj, first one. This is nucleus homogeneous positive sample. Everyone have seen? Anyone have not seen? Sir, you have not seen? Sir, how about you? Madam, how about you? Student, you can see that. I will show you in the this one also, projector also. Okay, nuclear homogeneous pattern which has come in this, which is a positive control. So you can see, you are not. Okay. So this is the liver substrate, no issues. Positive. Go to the negative one. Move it, Manoj, right side. Yeah. So we identify cytoplasmic pattern and other pattern in the sample which we received today. So this is the liver. This is a negative control which we have taken. There is no fluorescence available in this. So cell nucleus also we can't identify with this one. Yeah. Sir, there are two biochips. One is hep2 cell and another one is primate liver. Primate liver is animal liver. Okay, this is human cell. Human carcinoma cell which they made it grow, and as such this tissue is available all over the world. Okay. So hep2 cell is human cell. This is primate liver, animal cell. Yes, sir. That is the two biochip.
This is primate monkey liver cell.
Yes, primate monkey liver. Yeah. So positive, probably we need to go for some of the dilution in this also. Because if we are talking about this, we are showing the mixed pattern of cytoplasmic pattern also. Some of the nucleus homogeneous pattern also we are showing. So when we go for the next set of dilution, we will come to have the better clear results. But as of now, there are mixed patterns which is shown in this. Yeah. So again, nucleus is positive. Nucleus homogeneous we are seeing in this. And what we need to tell you one thing. So when we are not having proper incubation, sometimes when the water is available in this, and this kind of gap will come. So this also we will have to see. We will have one more session, not today, later, on troubleshooting. Okay, Manoj, you can check the next slide, probably. So you do one thing, show the first slide, they want to take the video. Yeah. Okay. Sir, once you complete the video, let me know. We are changing the slides. Yeah. So this is again it is a positive one. Take the better slide, the next slide. We'll see only one more slide here. Rest of the slides and other modern slide, like some of the positive slides I'm having with me. Some of the positive slides I'm having with me.
హలో హలో అన్న ఇక్కడ సౌండ్ ఎలాగరా సౌండ్ రావడం లేదు సార్
శివ ఏడ్రా
Second nuclear homogeneous one. But some of the we can see nucleus are positive. Yeah. It looks like a little pattern. Some of the kinetoclass kind of things are also. Yeah, kind of things are also there.
It's a homogeneous one.
Homogeneous one. Yeah.
Microscope in the bottom.
This endoclass to the cell plasma shading.
It's not kinetoclass. Originally, the pattern either it should be There's a center mid antibody, so pertaining to that it would be available. Know the next one. Okay, know the next one. I think you can go for the next slide. If it is not 5 minutes. Select the next slide. This is also this one. Somewhat positive is there. It is homogeneous. Right side, move it on the right side. Homogeneous. Yeah. Move the right one. So positive control will be almost same only. So this is the next one is negative control. How about other slides? So we have only 3 slides over here, or 5 slides. Yeah. Manoj, move it on the right side and adjust it. Okay, do one thing. Show this part. Show this one. Yeah, I'll show it on the big screen. Please wait. I'll show it on the big screen. Yeah, please come. Yeah, please wait over there. I'll show it on the big screen. Manoj. Manoj, please wait. Please take your seat. I'll show it on the big screen, please. Yeah. Laptop is there. Manoj, I'm removing the laptop. I'm removing the laptop. You can show them. Show the microscope. I'm sorry for the inconvenience. Majorly, we were having some patterns in the single slide which is not visible right now. Okay. I want to show you some of the other patterns which I am not getting right now. Maybe we'll be seeing the rest of the. Sir, I'll connect. We'll seeing the rest of the patterns, and as well as we can, have the. I'm running out of battery right now. So as we discussed, the cytoplasmic pattern, when it comes to the pattern number AC21, which is targeting on the mitochondria, where we can see the fluorescence on the cytoplasm, and as well as the mitochondrial cell also we can see the fluorescence, which is targeting M2, M3, and as well as M5 and M9 cells, which is focusing on the primary biliary cirrhosis pattern. Just a moment. Okay, I'm sorry again. See, mitochondrial fluorescence, when it comes to this, we can see the fluorescence on the cytoplasmic area, which is targeting for M2, M3, and M5, M9 antigen. Mainly it is focused for primary biliary cholangitis. The prevalence is up to 95%. The detection rate, we can use this pattern to find out that one. The next one, Golgi apparatus, which is available in the cytoplasmic pattern. It is focusing on the macroglobulin, Golgi 95, GM30, Golgi 160, and all those things. Majorly, it is focused for SLE and as well as Sjogren syndrome. We can see the fluorescence on Golgi apparatus, majorly focusing on the Golgi 160 and 97 and 245. In the similar way, if you are talking about the primate liver also, the hepatocyte is also stained with it. This is the next pattern. The rings and rods. Sir was not available, maybe he was telling about rings and rods. If you are talking about the rings and rods, we can see the filamentous structures expressed in all stages of the cell cycle, which is targeted for IMPDH2 antigen, which is associated with the hepatitis C infections. Majorly people who undergoes the treatment with the interferon alpha and ribavirin. So for those patients, it is useful. The last pattern, mitotic pattern. So in this mitotic pattern, majorly we are targeting pericentrin 99 and as well as CEP 110 and 250, and where we can identify 1 or 2 centrioles per cell. So we can identify the centriole fluorescence. So by this, the disease indicated is progressive systemic sclerosis. When we go for the dilution of 1:1000, we can find out Raynaud's syndrome also. So this is the next one. spindle fiber. When we are talking about the spindle fiber, we can see the fluorescence on the mitotic cells, but not on the cell nuclei, which is focused for Sjogren's syndrome and as well as systemic lupus erythematosus. The next one, pneuma. So when we are talking about pneuma, which is targeting pneuma antigen, which is indication is Sjogren's syndrome, SLE, and we can see the fine speckled and as well as reticular fluorescence on the nuclear matrix. Mid-body. So daughter cell and as well as the two daughter cell, in between the two daughter cells, we are seeing the fluorescence, which is focused for the mid-body, and other than that, it is focused for Raynaud's syndrome and malignoma and progressive systemic sclerosis also. So last pattern, MCA. The modified histone 3 MCA protein is targeted in that. In that pro and metaphase phase shows a dotted fluorescence. We can see the dotted fluorescence on the pro and metaphase, and which is mainly for polymyalgia rheumatica, and other than that, Sjogren's syndrome also, it will cover the entire thing. So these are all the 28 patterns which is coming under this one. So overall, when we want to go for ELISA method, prefer the method which is coated with the nucleosome in it. When you want to go for dsDNA, prefer the method which is coated with the dsDNA, which has the highest sensitivity and specificity, because the other method which is coated with the protamine sulfate only. The other method is coated with the protamine sulfate only, but with the uroamine it is coated with the nucleosome. These are all the other profiles are available, whatever the profiles we are running over there. So once we complete the immunofluorescence procedure, once we complete the antigen, when we want to go for the antibody to find out the confirmation, we have ANA profile 3 separately, and DFS and ANA profile MI2KU profile is also available. There, the 15 number of antigen SSB, SSA, and sclerosis 70, and PCNA, centromere protein, all those antigens are coated in this panel. And other than that, we have separate cytoplasmic pattern and ANA profile 23 also for confirmation. And this is profile specific profile. We have autoimmune inflammatory myopathy profile, systemic sclerosis profile, and autoimmune liver disease profile also we are having in it. So overall, to complete this, ANA is a gold standard to start with, and other than that, we have the biochip and diterplane technology, which ensure the specificity and the sensitivity in a better way. And other than that, the recommendation for the dilution, always go for the 2 dilution and come for the conclusion with the particular immunoblot procedure. And last but not least, the pattern which we are seeing, AC 1 to 28. We can identify the disease by using that one. Okay. Any other questions? Fine. We can go through the ANA pattern booklet which we have. Sir, switch on the lights. Sir. And we can see the procedure, immunoblot procedure, then we can close the day. Please come. We can see the immunoblot procedure. Making you to move here and there. Okay. See, based on the procedure, right now the 1.5 ml, the buffer is running right now. Sample buffer is running. That process is completed over here. We can see here. And other than that, the sample addition was done, followed by 3-time washing with the universal buffer is done with this procedure, and followed by addition of 1 ml conjugate from this also it is done. So right now secondary antibody is added with this procedure. So once the secondary body is added with this procedure, then they will go for the substrate addition. Once the substrate addition is completed, then final wash step will complete. So overall it takes another 1 minute to complete this process. To complete this process, the other rest of the process, it may take another 15 minutes. So after that, the result will be evaluated. Okay, so majorly, see the profile which we are running right now is ANA DFS only. Okay, so the positive case which came for ANA, so we will be finding the band in it. So we can measure whether it is 2 plus or 3 plus positive. So based on that, we can report the patient. Then they can go proceed for the next step. Okay, so when you want to go for the monospecific ELISA, when you want to check the particular antigen, then you can go for the ELISA. Okay. Completed. It will come. I think they need to move there. Let them go. Once it is completed, then we'll show the procedure. It takes another 15 minutes to complete. Yeah. Yeah. Okay. See, overall, I want to tell you only one thing. So whenever it comes to this IF and pattern, it is a easy procedure. When we are doing this procedure in a continuous way, we can complete the procedure in a better way. That's the first thing. Second thing, it is a gold standard in identifying the antibodies, which is again the autoimmune disorders. So which we can, complete by diagnose by using our Euroimmune kits. And other than that, for confirmation, we can use the blot or ELISA method, which is a procedure which we need to follow. And some other things will happen. For example, some other people, when they use the tray repeatedly, then the band will become black. So, and other than that, proper drying, without drying the strips also, if we scan, then the negative results will come. So those are all the things are there. But within this day session, I can't take everything. But right now, we learned about ANA, and as well as we learned about what are all the patterns are available. We are not gone deep into the patterns right now. Maybe the antibodies, some of the antibodies which is essential for this, we have seen that, and as well as what are all the procedure for IF we have learned, and other than that, immunoblot procedure also we have learned. Okay, so that's it from my side. Any questions? ELISA DNA is available. Extracted is monospecific ELISA is available. Extracted is not available. Okay. Yes. See, once we complete the procedure, we are completing the procedure in 15 minutes. Once we complete that, what we will do, we will remove the strip. We will take the strips out. We will dry with the normal tissue paper. Then we will scan it over the scanner. By using Euroline software, when we are scanning it over the scanner, the band quantity, based on the band intensity, the antibody level will be measured. So it will come as a report. For example, no, once you will have to scan the strips over this one, then it will come to the monitor by using this one. Let's see, these are all the report copies available. Okay. It will come as PDF. You can see the report. Yeah. So this is the way. Please come forward. You can watch this. I'll zoom it. Yeah. So patient name, created date, and all those things, strip number. These are all the antigen, which is known for allergy test. In case if it is for ANA test, the ANA antigen will come, concentration, and the classes will come in plus category. So the result interpretation will be given in a single page itself. So antigen and positive or negative, what is the class? All those things will come in this. For allergy, it is coming this way. Okay, so this is for allergy, we are following East class, 0 to 6. So 0 means there is no antibody. 1 means very weak antibody. 2 means strong antibody. 3 is very strong antibody. 4, 5, 6, it is very, very strong antibody. So, he is allergic to rugweed and as well as mugwort. So the band, it has crossed 3. So that means they are allergic to particular allergen. So for him, these 2 allergens are positive. And then control band also, as I told you, it should cross the number 3. So it has also crossed number 3. Serum. Blood serum we have taken. And it has crossed the 5. The test is also valid, and as well as the patient was actually allergic to common rugweed and as well as mugwort. Alternator. These are all molds. These are all the allergen. Basically, I am showing the allergy profile right now. So, yes, sir. Once it is done on the ANA, so we'll be having ANA antigen over here. It will come as single positive or borderline positive or 3+, all those things will come. We can analyze based on that, which antigen is positive against it. Yeah. See, for example, here the antibody concentration is 80.95, 80.85. So here it has crossed 3, and as well as it has reached here, then there definitely the patient is allergic to these 2 allergens. Yes. For example, this antigen, they are not allergic to this. They don't have that much antibody, specific antibody against that particular antigen. Yes, yes. The similar way, whatever the antibodies we are having, SSA, SSB, all those antibodies will come in ANA report, but ANA we are not giving the quantification, we'll give as a single positive or double positive, borderline positive or triple positive it will come. So strong positive, 3 positive, 2+ will be considered as strong positive. In cytoplasmic profile also. Oh, that's not my mobile. It's not my. Okay. Madam, any other questions, sir? Okay. I'll show you, sir, once it is completed. Because allergy profiles, it is all those things are available. ANA, I will have to get it from them. Yeah. Okay. For ANA also it is coming in the same way, like antigens are available, as I told you. Antigens are available, intensity is available, and classes are available. As I told you, you can say the class as single positive, double positive, or borderline positive. All those things will report as semi-quantitative or qualitative. Yes, sir. How many? Dilution. For example, if it is a ANA, if it is 1:100, ANCA and other thing, we need to start with a 1:10. So usually, once it reaches the titer, 1 is the weakest one, we can go till that. But the thing is that take whether it is the strongest one or weakest one, based on the fluorescence only. By seeing the fluorescence visualization, we will come to know that. Yes, sir.
By starting from the dilution, we will do continuously diluted.
Excuse me. Please listen, please. Kindly pay your attention. Excuse me, sir. Excuse me, sir. Only 1 minute. Only 1 minute. Sir, 1 minute, sir. 1 minute. Sir is asking that for dilution, how I will come to know the positivity. So that is the reason. As a standard dilution, we are recommending for ANA 1:100. For ANCA, start with a 1:10. If you are getting the strong fluorescence, if you want to find out the titer, increase the dilution. Then you will find out. Suppose if you are unable to see the fluorescence in that, then increase the dilution. You will come to know in which point it is coming. So always we recommend start with 1:100 for ANA and 1:10 for ANCA, and later still if you are not having the fluorescence clearly or higher fluorescence, when you want to find out the titer, go to the next level of dilution. So 2 dilution would be sufficient, sir. Start with the 1:100 or 1:10. Sir, you are asking something. It is a software. Excuse me, one more question. One more question. Sir is asking that, see, once we complete the test, we are measuring this by using the strip. Then which method we are measuring the strip? Okay. So this is a Euroline software which was given by Euroimmune directly. So how they are doing? They are measuring the intensity of the bond. So this intensity of the bond, it indicates the quantity of the antibody. So the intensity is equivalent to quantity of the antibody. When we are scanning this one, the band of intensity of the band is more, means the more number of antibodies bound in that. So that is measuring the intensity. Through that it is measuring the quantity of the antibody. So if we have lesser number of the antibody, the intensity will be lesser. The intensity is equivalent to quantity of the antibody.
Now, what is the substrate you have added? Type of substrate you have added?
It is a coloring agent which we have added. It is NBT BCIP. NBT BCIP. N-butane.
Like OPD, like that. TMB OPD. Biotic labeled for అందులో LP.
Sir, we have used NBT only. I will tell you what is the exact substrate which is used for coloring agent, for coloring. Okay. Hope many of you are tired. I am also tired. Have your tea. And if anything related to product queries, we have our person, and as well as you can visit our website. It is Everlife CPC Diagnostics. You can check that. And we have a person, Mr. Manoj, who is available here. Maybe he will give you a card to you. He'll contact us. Okay. Thank you. Thank you all.